So ultimately where we ended Monday was that we're talking about the electrophile additions, right?
Yeah.
And our electrophile additions center on non-polar pi bonds. Okay, so that's our primary structure. Almost everything we'll look at is alkenes. But officially the alkynes do very, very, very similar chemistry, if not identical. They've got some kind of secondary things that can make them a little bit more complicated, but ultimately it's the same functional group, it's the same chemistry. All of those reactions end up running the same. And so one of the things that I pushed was that you're responsible for pretty much all of these reactions. Okay? And what we're going to look at in a little bit is how would we use this diagram by looking at some of the practice questions from that packet that you've probably actually already worked, I'm going to be honest. But then we also get these reactions, and we're looking at our electrophile additions. Play posits, did you get a chance to watch them all?
Yes.
Yeah. Did you have any questions specific to them?
No, at this time, no.
No, that's fine.
But still need to repeat watching it.
Yeah. I would argue there's a lot of information in it. This first one, if I'm correct, this first one is the more straightforward of the two. And then the second one, I fully admit, is challenging. And what that second one looks at is the electrophile addition as opposed to the acid addition. Both of them still fit under the electrophilic addition type classification. But the acid addition is a little bit more straightforward because it uses acids, and that helps drive a lot of that chemistry. So when we look at that acid addition, we have a reagent we're familiar with. Okay? That reagent is usually a strong acid. So when we're looking at it, we're looking at something like HCl. So HCl being a strong acid, we know donates H+. So we can see this and say that's H+. If that's H+, this has to supply the electrons. That helps us kind of drive some of that chemistry. Okay? The other big thing to kind of watch out for when we look at these is a lot of this chemistry is going to be dictated heavily based off of the starting structure we look at. So if we think about what I drew up on Monday, we just looked at a single nonpolar pi bond. Both of these carbons in that pi bond are equivalent. So it doesn't matter where we would start putting our electrophilic atom. It could go to either carbon. If we look at the carbon structure chosen for this plate posit, it's a little bit different. It's not perfectly symmetric. And because it's not perfectly symmetric, there's now a preference on where the hydrogen should go. Okay? And at least our first step. The cheat that we can get through is remember that we have resonance. So there's really two resonance options that we could try to do. We could take the electrons in that pi bond, and we could move them to that carbon, in which case we end up with the structure like this, with the negative charge there. And now where's the positive? We'd have a positive charge up here. Okay, so knowing that resonance can help us guide the chemistry of our molecule. Okay, if we were weak with resonance, everything falls apart and you now have to memorize something else. Or we could have moved the electrons there. So now there's two possibilities. So if there's two possibilities, then shouldn't we get multiple products? Couldn't the H, our positive hydrogen, react with the negative at the terminal carbon versus the internal? Theoretically, yes. Okay? And that's going to make things a whole heck of a lot more complicated. Except that's only true if they're equally stable. Is a primary anion equally stable to a secondary anion? And hopefully we know, based off of our last exam where we had a question on it, right? In fact, 2 exams, that the primary anion is more stable than the secondary anion, which means when we consider these two resonance structures, only the blue one is relevant. which means the blue one's going to dictate where the chemistry goes. The hydrogen, being positive, is going to connect to that negative. There's the answer. I don't see the situation where the hydrogen's connected in the other spot because those charges wouldn't align. Okay? So I can find the positives and negatives, evaluate if there's asymmetry within the molecule, and start to predict where those things align and come out, which is kind of a neat system if we can leverage that old content. If we can't leverage that old content, we need something else to cheat our way through it. Okay? And that cheat our way through it is why did I not get this system? Okay? Well, the cheat that you'll typically hear reference to is what's known as Markovnikov's rule. Okay? The hydrogen in an acid addition goes to the position that has more hydrogens. Which carbon has more hydrogens? This top one only has one hydrogen. This bottom one has two. So I put the hydrogen at the bottom position. And that becomes the answer. Okay? And that one becomes an invalid answer. That requires me having to memorize this extra rule. What is the point of that extra rule? It's utter nonsense. It's a way to try and cheat the chemistry so that you don't have to learn the chemistry. The problem with that rule is it fails. And there are plenty of scenarios where that rule doesn't work, which is when people go, oh, well, we just won't call it where the hydrogen goes. We'll now call it, it's anti that rule. I mean, you're having to memorize even more things to explain something instead of actually explaining it. Yeah, that's the problem that I've got with it. That phrasing that we're hearing here is what's known as Markovnikov's rule. Okay, Markovnikov's rule is great. It explains where a hydrogen goes in a very simple system. You make the system more complicated, everything goes out the door, and people have a hard time processing where all those pieces go. We switch to very related chemistry. Electrophile addition, instead of using acids, use a strict electrophile. And now we have to memorize a whole new set of chemistry, except hydrogen was positive, it went to the negative atom. The electrophile is positive, it goes to the negative atom. You mean the chemistry is identical? Yeah, it is. So if we focus on the chemistry, we don't have to memorize some nonsense rule. Why do people push the Markovnikov rule? Because it's easier to tell people to memorize a fact than it is to explain that fact. Okay? And that's why those videos show up all over the internet, as people go, oh, you got to do Markovnikov's rule. And what they're effectively telling you to do is to memorize the answer to the question. Okay? That only works when you know the question. And that becomes the major issue. And that's why organic chemistry becomes a challenge, is because students are trying to memorize their way out instead of understand it. The rest of the play posit goes through and looks at a variety of different examples, both the alkene and the alkyne. We can look at another system where we look at sulfuric acid, which does the exact same thing. And again, people will panic and freak out and say it's doing something else, except it's doing the exact same thing. It's adding H+ and a nucleophile, in this case OH. That creates a new functional group. That new functional group is unstable and reverts to a different functional group. The chemistry of the reaction maintains the same. Okay? The next part of the video goes through and says, well, what if we have conjugated pi bonds? Because conjugated pi bonds add a wrinkle to this. Because with more pi bonds, you get the potential for more resonance. More resonance means more complexities that you have to think about as you go through and process the reaction. But ultimately, it's the same chemistry all the way through. Okay. This then goes through and addresses, that ends with, I think it should end a little bit more on a somewhat cliffhanger or odd reactions. If we can change what materials we're starting with, we can get some odd responses. And the odd response we get if we realign that thing is we can get this cyclic structure. This is a very famous named reaction turned into Nobel Prizes. Okay, it's known as the Diels-Alder reaction. It's cool, it's fun, it's neat, and it's probably outside the scope of this class, so I wouldn't stress too much about it. The next one goes through and looks, again, trying to boil down the basics and repackage it and respin it. Okay? But in this case, what we're going to try and do is look mostly at the electrophiles. So it reviews the acid-base reaction, looks at some energy diagrams behind it, addresses the Markovnikov's issue again, addresses some stereochemical traps, which is slightly different than we had in the last one. And then we get this part, which I think is still in that same video. It might be a third video. Okay? And it looks as the pi bonds acting as nucleophiles instead of the pi bonds acting as bases. When the pi bonds go act as nucleophiles, you get a whole new class of reagents. Okay? You get this system where you have a Lewis acid, something that accepts electrons, and some kind of leaving group. There's a whole ton of reagents that sit in this. Okay? Those reagents are really hard to classify according to their mechanism, and no textbook seems to do a particularly good job of organizing it. I don't like lack of organization within a system, so I kept staring at it, and what I noticed was a certain pattern that started to fall out. If you go through and look at that thing that it's reacting with, if you have a good leaving group, you end up with a 3-membered intermediate when that mechanism comes together. Oof. There's that 3-membered intermediate. Okay? Good leaving group, you get this weird 3-membered intermediate. Okay? What happens if you have a bad leaving group? Well, if you get a bad leaving group, you get larger structures. You'll get 4-membered or larger ring structures. Okay, 4, 5, 6-membered rings. Okay, but that requires looking at our reagents and finding those patterns. The rest of the video attempts to make you go through and look at these structures, look at these reagents and say, how could it act? Could it act in a scenario where it would accept electrons? If it accepts electrons, what would the leaving group be? How could it leave? And try and pushing through each of those individual systems and leverage being able to compare and look at the reactivity of an individual structure. Okay? So you get a variety of different systems to go through and explain what's happening within those. You'll note even within the generic system, you get these question marks. Those question marks have to do with whether it was unstable. If you form an unstable structure, yeah, it's going to continue to react. But if your product is stable, it stops. So you'd have to look at your individual structure to decide what happened with those particular reagents. So it's trying to group and come up with general categories without having to go through and memorize a ton of reagent systems behind it. Okay? That is challenging because it's trying to generalize a system to be able to apply to very large specifics. That's hard to go through and do. It takes practice. You don't get a whole lot of practice, unfortunately. Okay? So we got to do the best that we can within the system we've got, and we'll go through and talk through some of that in a second. For those people being like, well, why are you going so fast through all this? This is all PlayPosit. You're supposed to have watched it online. We're really just kind of calling out questions. These last couple slides are things I added freshly this morning. And I'm finished playing with the slides for the record. So if you go through and look at all of our slides, you'll have a couple new slides for, I think it's a total of 6 new slides within this deck. So since you've got them printed out, I apologize. But okay. Most of the slides really just have a structure and some images. So one of the things we typically get when we talk about chemistry or any of these things is, why do we care? Okay, so why do we care about nonpolar pi bonds? Well, nonpolar pi bonds have higher energy electrons in the pi bond. Okay? And those high energy electrons are what do all the chemistry for us. Okay, so that's doing all the chemical reactions. They also can be used to do not chemistry things. Okay, so if we think about atomic line spectra back in 130, you could excite electrons in an atom up to a higher energy level, and when they crashed back down, they emitted light. So if you took, or theoretically everybody took the, what are they called? Yeah, you at least know what I'm talking about, but you're running into the same issue I have, the spectra things.
I think it really was called like
It's something, yeah.
spectral.
Maybe a spectrometer. I'm a little nervous about calling it a spectrometer. But sometimes it can just be a diffusion grating, so it's just a clear sheet of plastic and you hold it up to the light, and if you look at it, you'll see lines, right? Or if it's white light, you'll see a rainbow. If you hold it up to the sun, you'd see a rainbow spectrum coming off of it. Okay. What those are looking at is electrons moving orbitals. That's it. Jumping from a low energy state to a high energy state and crashing back down. Okay. If you've ever been to or heard of blacklight parties or walked around during Halloween and seen like shirts glow, that's the same process. The black light is being used to excite a particular electron in the clothing you're wearing to jump to a higher energy level. When it comes back down, what happens? It emits the glow of that light. Okay? Not all colors do it. Why? Because that gap between the high and low energy states is tuned to particular energies. And if you change the color of your material, you're changing the energy of that gap. That's why it doesn't work for some colors and it does for others. Okay? That energy is tuned by changing what is known as the conjugation of the pi bonds. Conjugation is a fun way of saying resonance. Pi, sigma, pi, sigma, pi, sigma, pi, sigma, pi, sigma, pi. All of those double bonds are in conjugation with each other. I can move electrons from this carbon all the way out to this oxygen and vice versa. That's pretty powerful. And by doing that, that shrinks the energy gap between the highest and lowest energy electrons, or orbitals, I should say. And that changes the light with what the molecule gets absorbed. Okay? So it's kind of cool. Oh, we could tie it back to blacklight parties. Yeah. But why else do we care? Does anybody recognize that molecule? It's retinal. Retinal is typically found associated with eyes. Okay? This is why you have retinas. Okay? What's the point? Well, what do eyes do?
They filter light. It's like, it's a mirror.
Reflection?
Yeah.
They absorb light to do what?
To see.
Yeah, real simple, to see. How does that work? Light comes in, hits a pi bond, and it breaks the pi bond. So what happens if that pi bond breaks? Should have picked a different pi bond. What is now possible?
Resonance?
Yeah, there's some resonance. Is there as much resonance? No, why not?
Because you're missing that pi.
Yeah, we're missing the ability to pass through that. What is that? What did I point to?
Sigma bond.
Cool. What's special about the sigma bond? What's that?
Cannot break.
Yeah, they're harder to break. We can still break them. That's all the chemical reactions we've done. How are sigma bonds different from pi bonds?
It's a different hybridization of.
True. Why does that matter?
Like your different levels, you can't move the electrons around the same way.
serve
Why does that matter?
different orbitals that don't overlap.
Why does that matter? Maybe a hint would be, what am I doing?
Orbiting.
I'm not orbiting anything, though. I'm whatting?
Rotating.
Rotating. What can a sigma bond do? It can rotate. Why can't a pi bond rotate? Because the orbitals overlap, preventing the rotation. Sigma bond has a direct overlap of orbitals, which means I can rotate and it doesn't change the overlap. Why does that matter? Damn it, I'm really dizzy now. 1, 2, 3, 4. Dang it. I can do a rotation. Why does that matter? What is different about this structure like this versus the structure like this? Okay, with the hands, what is the difference between the structure like this, head, straight body, versus the structure like this?
It's just positioned differently.
Why does that matter? You're standing around with friends, right? You're standing around, probably not a big deal, right? But if one friend does this to you, what are you going to do?
Kick them in the butt.
Wow, that was a pretty aggressive change. All they did was bent over and now you're going to kick them? Why?
It's different reactions. It interacts differently.
It interacts differently with its environment. That's really neat. How do our eyes see? We have a whole bunch of these straight chains. They absorb light. All of a sudden, that molecule bends. That's now a physical change within our eyes. What does our body then decide? Oh, physical change means light and of this particular energy hit me at this particular pixel within my eye. Which means I need to tell my brain at this pixel point, I have this color hitting me. You just created vision. Now build out an entire spectrum of these potential molecules at each of your little individual pixel points within your eyeball. And you now have the capacity for light or vision. That's pretty wicked. And all that's happened is you rotated a bond. Very subtle change, and you get huge implications for what we as humans perceive in the world. That's pretty wicked. Right? What else could we do with it? Oops, I forgot that button doesn't work. So still pi bonds in light. So we got a bunch of pi bonds here. We've added some different types of pi bonds in here, but for the most part, it's still pi bonds in light. Anybody know anything about these molecules? I know we were all nutritionists and not skincare experts. Does that help anybody? Nothing about skincare?
Is paraffin?
That's not paraffin, no.
Paraffin.
Uh,
Never mind, doesn't matter.
I don't know enough about the nomenclature of that to be able to answer that question. It is possible that those are rooted in each other. I would bet at one point or another in your life you've put one or more of these compounds all over your body
Is it sunscreen?
as sunscreen.
Chemicals go with sunscreen because it absorbs the
Yeah,
UV and then
it
reflects
absorbs the UV light instead of your skin. Okay? So sunscreens take advantage of pi bonds because pi bonds interact with light. So you can put a sunblock or a sunscreen on your skin, and what it does is absorb that UV radiation instead of your skin. Because if your skin absorbs it, it turns it into chemically reactive things that can cause degradation of your skin. This is why you can get blisters, because you end up killing enough of the skin surface that it's like, oh, I'm dead, I have to protect myself, and it protects itself by creating blisters. Yay! Isn't that lovely? But these molecules' sole responsibility is to absorb UV light. And that requires pi bonds. Right? There are other forms of sunscreen out there too, like you can use mineral ones, and what do the mineral ones do?
Deflects.
They're usually deflecting the light. Those become a little bit less popular because what happens to your skin when you put a mineral sunblock on?
It's white cast.
White cast? Which might, I don't know enough about the words.
Isn't that like where you get the, like, you know, like the zinc or whatever you can put on your nose? It's
Looks ghostly, okay, that's the
Very,
white
yeah.
cast. It's because it's literally preventing, you're painting
Creating
your
a barrier.
face, creating a physical barrier between the UV light and your face. Okay? But if you put a physical barrier on, there's something blocking all of the light coming out of your body as well. Okay? Or the light reflecting off of your body. So you look different. Okay? So you change how people perceive you just because of how that material is working. All of that is chemical based, and it's deciding which chemical is potentially healthier or better, right? Or more ideal for your particular scenario. One of the things that we continue to find is that in these processes, we almost always create molecules that have some kind of cancer effect. Okay? And a lot of what that cancer effect has to do is these structures are benzenes, because those benzenes are flat structures. How much do you know about the structure of DNA? Double helix, cool. So we could pretend to draw a double helix, because, you know, why not, right? I can... You just laughed at me. That was harsh. Okay. What's that?
So it goes the other way. Yeah, I feel like
Fine, you stinking biologist.
it doesn't matter. Yeah, that looks.
Well, that looks a little better. Yeah. Okay. Just needs to be a little bit better. Is that it? Just a double helix?
Chains.
Okay. With the nucleotides in between, okay, not RNA, DNA, they're deoxynucleotides. Okay. How does that hold together? Actually, let's scratch that question. See each of those rungs? What's in that space? What's in that white space? It is fluid. There's pretty much nothing sitting in that white space. What do need to go back to it. What's holding the DNA together? Why are the strands two strands? How are they held together? The nucleic acids. So we could put blue dots in between, right? So this is really two strips, so we could unwind it, right? We'd have this, more or less. And the other side, we'd have this. And it's when we connect those. So what holds them together?
A bond.
And this becomes fun, the polypeptide bond. The polypeptide bond and all the bonds holding these together hold this together, and they hold this together. They do not hold this together. There is no chemical bond between them. What holds them together? So using the emphasis. Okay, yeah, they're intermolecular forces. Okay, and typically We pair C with
G.
G, T with A, X with... I was just testing you, making it up. Okay. So why does C go with G?
That's what they're compatible with.
What's the compatibility?
I don't know. That's just what I learned.
They're, isn't it just the way that it lines up,
What's lining up?
your helixes?
The amino acids. No,
They aren't amino
they make
acids.
up an amino acid
They
after
don't do
you
that either.
decode.
Yeah, later information.
Something like substrate match with.
Hydrogen bonding. There's hydrogen bonding between the individual amino acids. You got me doing it now. Hydrogen bonding between the individual nucleotides is the word you were fishing for before, and that's why C bonds to G. Okay? But that's why C bonds to G. Why does DNA hold together as a double helix? It's vastly more complicated than what I'm about to describe, but this is a missing factor in most conversations. Everyone goes, oh, it's just the hydrogen bonding between the two chains, and that's not true at all. One of the largest contributing factors to these structures holding together, and particularly chaining and intertwining like this, is that the individual nucleotides are rings. I'm going to pretend I can draw a nucleotide. I don't know, right? We got some other nucleotides, except it's a 5-membered ring, isn't it? I didn't think to look up nucleotides before lecture. I apologize. You got nitrogens within this, a double bond. So you can get hydrogen bonding. Oops, maybe there's an H there. You can get hydrogen bonding between those. Maybe there's an NH here. You get hydrogen bonding there. Vaguely familiar? Okay. It's not those hydrogen bonding interactions. It's that these structures have a whole bunch of conjugated pi systems within them. Those conjugated pi systems like to stack on top of each other. That's what's creating the strong kind of attraction between these, is that we can neutralize some of those interactions by stacking them. Which means, what else could I insert into that space? How about other ring structures? And as soon as I slide another little ring structure into there, what happens? That space opens up a little bit, and it creates a kink within the DNA strand. What happens if you have a kink within the DNA strand? A mutation. Now when the rest of your enzymes or proteins come in to read the DNA, there's a kink, and it goes, oh, something's wrong. Okay? So it tries to edit it, and it opens up the DNA strand to edit and fix that. But every time you make an edit, what do you have the possibility of happening? A mutation. Right? And yes, DNA replicates insanely well. Right? But over time, mutations accumulate. It's called aging and ultimately death. Okay? Mutations do occur. And if we're starting to add molecules that fit into that space, we increase the amount of times that that editing process has to happen. And if we increase the amount of editing processes that happen, we increase the odds for a mutation to occur, and that mutation can be very detrimental to the existence of whatever that species was. Okay? So our sunscreens do a great job absorbing light, but at the same time have this cancer-causing effect because they can insert themselves into DNA. Neat. Yay, pi bonds. Okay, cool, we can see things and they kill us. So you can see that you're dying. You gotta love it. Okay, what else can we do with pi bonds? You heard the terms saturated and unsaturated before?
Fat.
Yeah, okay. So we could look at saturated fats and unsaturated fats. Why might I lose my crap at this picture.
Because it's a lot. It's a long chain.
True. Why else might I have some problems with this drawing of this picture? Octet's fine.
It's very bulky.
What's the shape in here?
90 degrees.
Yeah, what shape is it supposed to be?
Zigzag.
Yeah, it should be a zigzag. So if we're going to look at this a little bit more appropriately, we should be seeing a zigzag. Even within this zigzag, I'm already drawing a problem, okay, at that point, but we're just going to kind of pretend and ignore that. What happens with the bottom one?
Does it curve onto itself?
I'm still going to get a zigzag, but at some point I get a double bond. How is that double bond going to affect my structure?
Are you gonna make it go up?
What's the bond angle above?
Not 90 degrees.
And that's actually a fair statement because I don't think I told you to memorize bond angles. That shape is ultimately tetrahedral, and that gets us 109.5 degrees. What's the bond angle here? Let's go with shape first. What's the shape? Trigonal planar. That's 120 degrees. If we continue this chain up, we can go this direction. Is that going to change how those two fats interact with their environment?
Yeah.
Yeah, you've got one that's a straight chain and one that has this kink in it. Okay? And when we consider fats, it's not a single molecule, it's billions upon billions of these molecules. And if I look at stacking a whole bunch of saturated fats on top of each other, they're going to stack relatively nicely, all nice and neat straight chains, more or less. Which means lots of interactions, which means its melting point should be,
High.
let's pick something high, okay? What happens when we look at the unsaturated fat? Will I be able to stack all of these nice and neatly on top of each other?
No.
No, which means the intermolecular forces get disrupted, which means what happens?
They're weaker. Right?
Say again?
They're
Weaker forces, which means what happens to its melting point?
weaker.
It gets lower. Why is this particularly relevant for us? Where have you seen saturated fats?
Oil
Careful.
and food.
True. Where do you typically see saturated fats? We've got some nutritionists here.
Mostly see them in the ingredients list or a nutrition label, but I mean, or
Where are they coming from to get onto that nutrition label?
Like processing?
Yeah, where did it come from? Where did we pull, where did we source? Because I don't go to the store and say, give me saturated fat.
Plain,
Say again?
plain.
Plain?
Seed.
Okay, plants and seeds do have a source of fats. You're absolutely right. Okay, are they saturated fats or are they unsaturated fats?
Unsaturated because it's natural.
Everything's natural, more or less, so you gotta be a little careful with that statement. Okay, your plants and seeds almost always universally have unsaturated fats. Okay, and because they're unsaturated, they don't pack neatly, which means what happens to their phase?
losing.
They loosen.
They're liquid.
And if you consider all of your unsaturated fats, your olive oils, okay, your canola oils, your seed oils, what phase are they all? Liquids. Why? They got a kink in them, preventing them from stacking appropriately. So where do I find saturated fats?
Butter.
Process.
Dairy.
Butter, which is coming from
Animal.
animals. If we look at animal fats, almost all of them are saturated. They're in the solid form. They're able to pack better because they don't have that unsaturation through them. Right? The question of why do animals have that and plants don't, I don't have that answer. Sorry, I didn't study biology enough to know that answer. Right? But I do know that that connection is there. Right? And that's why, if we think back 50 years, we're like, oh, butter, saturated fat is killing people because it's clogging people's arteries. The saturated fat is sitting in there and turning into lard balls and preventing people from allowing blood to pass through. So how do we make people healthy?
Unsaturated fat.
We go to unsaturated fats. Okay, but what's our primary source for saturated fats? Where do we usually find them? Animals. Butter. You're like, well, but I want to be able to spread my butter on my toast. Well, animal fat is bad then. You can't use animal fat. But what if we take plant fat? Plant fat is now a liquid. So we'll just give everybody oil. You just pour it on. Oh, I don't want to pour it. I want to spread it. Okay, well, how do we get it to spread? What do we have to do to it?
Chill it.
Sometimes, yeah.
Like water.
You start to saturate it. You hydrogenate it. You add hydrogen. You add hydrogen to convert the alkene into an alkane. So we get the I can't believe it's not butter, which was plant fat, so it's healthy, right? It's the exact same chemical that you're producing, saturated fats. You're just producing it chemically as opposed to, or man-made chemically, as opposed to just letting it come from the animal versus the plant originally. Right? So this is where language can become a problem. You go, oh, animal fats are bad. Plant fats are good. So we'll take plant fats and turn them into animal fats, and therefore they'll be good because they came from plants. That's not how it works. The chemical structure is identical. The chemical structure is identical. You get the exact same issues either way. For the record, you're not forming little balls of fat in your blood. That way, at least. Okay? Which is ultimately why what has typically happened to that hydrogenated fat phase just kind of disappeared because we realized we're now doing more processing, and that's less good. Okay? So what does this mean for us as far as lipids? Okay? Well, we can get our alkenes. What's the issue I have within this? We've lost information about shape. Right? And then if we look at, which is fun, because the whole point of this is a difference in shape, and yet they're not showing shape at all. And then they half-ass it down here. To show, oh, it's bent. And, well, if you're going to show shape, show shape. That's like me going into a school to be like, here's a triangle, kids. Because there's a straight here, and there's a straight here, and here's a square, kids. Right? I've got a square because I've got 4 edges on this. If you want to show a square, show a square. You want to try and show something that has shape, show a proper shape. Okay? What happens with these? Unfortunately, all this really is so far are a fatty acid. And what happens in biology is we scaffold these things up into larger structures. We add molecules known as glycerols. Okay? And we would end up getting lipids. Well, where did our fatty acid go into each of these lipids? Well, there's a carbon, there's an oxygen, and an oxygen. That's the COO. And this R is that chain. Okay? And then it's connected to this carbon structure in between. And if we go through and see, we've got that COO there with another R. This last one gets a little bit weird, so we'll come back to it in a second. But we get this core functionality through it. That core functionality is what's known as... You want to repeat that again, Lindsay?
So,
Close. I thought you said something else. Glycerol?
yeah.
Okay. Glycerol is the thing in the backbone that holds all the fatty acids together. Once we cleave the fatty acids off of that, here we have our, we'll call it natural, that says natural, soap molecules. Okay, so that's what we went through and made a while back. So we took one of these large lipid structures and we reacted it so that the glycerol is cleaved separate away from the fatty acids. Those fatty acids in their basic form, where they're negatively charged, are soap molecules. Right? And when we link these now to larger structures, number one, it can bring them more together and helps group them a little bit better than just having them free floating off on their own. But then we can also, because of the glycerol, sacrifice one spot to change some of the end chemistry of that molecule. Okay? So if we go through and look at these, what we're doing is modifying that end piece. That end piece, depending on its charge state, would typically, God, this one looks weird. Oh, sphingomyelin went real wicked weird on us. 1, 2, 3. I think there's our glycerol, more or less. Those things in purple now can modify some of that secondary chemistry. Right, so we could arrange the lipids now with a whole bunch of them into a phospholipid bilayer. We call it a phospholipid bilayer. Why do we call it a phospholipid bilayer? Phospho for? Phosphorus. So when we link a bunch of these together, we get a phospholipid bilayer. And because I can modify what's connected to that phosphorus, I can now modify how that part of the cell wall interacts with the outside or inside parts of the cell wall. Annie, can you help me out? How do we determine A, B, or O blood type?
How to figure it out?
Mm-hmm.
We add anti-cell.
Mm-hmm.
Yeah. Actually, we do two type, the forward type and reverse type. So the forward, we add the red blood cell suspension with anti-A, anti-B, and anti-D. So, for example.
What are those anti-A's interacting with?
Antibodies.
What's the antibody? What's it
Yeah.
responding to?
The antibodies in the
React with
Found
red
where?
cell. The red cell surface.
On the red cell surface. You mean it's interacting with a tag, a marker on the outside of the red blood cell? Yeah. There's something hanging off the red blood cell. That thing hanging off had to be connected to the lipid surface somehow. Is it connected through a phospholipid? I don't know that. Okay, and I don't know if you know that either. Okay? But there's something on the surface of the red blood cell that our anti-A, anti-B are responding to. And when it links up, because it sees it, it changes color.
It's glum.
It starts to clump. Okay. And because it's clumping, that's a now visual response or a visual cue for us to be able to say, that means the red blood cells supplied a thing that responded to that. They responded by looking for one of these signals on the outside. So there's a ton of different things you could link on to that purple end. And potentially one of those things could be the markers for A, B, or O. O means nothing, right?
Yeah.
Yeah, so either A or B blood typing. Okay, so it would allow us to look for that. There's also, was it the Reese's? Is that right?
The R.
The RH.
We test with anti-D.
And what is RH? Is it Reese?
Yeah. No.
Or Reese's?
Research.
Okay, yeah. I never know how to say it, and I keep calling it the friggin' peanut butter cup. Okay. But it's following a similar process. So understanding these structures can allow us to then manipulate and say, well, where are these things coming from? Why do these things exist? And so a lot of useful things can come out of it, but at the same time, a lot of bad things can come out of it too. Just looking back at margarine versus animal fats, okay, versus butter. Okay? That's a very subtle change, and the reason we're using margarine is just to solidify it. The whole reason we were getting away from animal fats and butter was because it was solid. By just converting your plant fats back down doesn't help anything. It just gives you a different way to source the exact same chemical that could be causing problems. So what we want to do is think about what's happening structurally within these pieces instead of just saying where they came from. What are they chemically, and what do those things mean? Right? That is challenging. No one person probably, or I would argue no one person should have the ability to know all of, well, it would be nice if everybody had the ability to, but I doubt any one person has the ability to process all of that content because they have to be an insane expert in all of those different levels. One of the reasons you're responsible for taking a class like this is to pick up some of those pieces so that if it boils down to it later on in your career path, you have the ability to evaluate and potentially call people out like weren't called out for the hydrogenated fat phase. Kind of makes sense? That's it as far as alkenes go, in my opinion. Which takes us to where I would like to go. So now a little bit of a mix mash here. Would be what can we go through and do with tests? So number one, we have covered now some structures about lipids, their properties, are fat saturated and unsaturated, and how those things boil down. I don't have any practice questions on that. Ultimately, they're just facts and applications of the stuff we just walked through. And of course it never loaded. So if we go to the practice, what is the most likely product for the reaction below? So what do you see? So the first thing I would argue you should do is look here. What do you see? What?
Triple.
Right. Hopefully what you see is the triple bond. You might be, well, what about the aromatic ring? Didn't we start talking a little bit about those? We did, but one of the things that I think most people missed before we walked in is that when we're talking about our electrophile addition, we're talking about alkenes and alkynes. We do not talk about aromatic rings. How are aromatic rings different from alkenes and alkynes? They are very much more stabilized due to cyclic resonance within that system. That takes the individual double bonds within an aromatic ring and effectively makes them a lower level alkene. Lower level enough that they become unreactive. Okay? Very end of the semester, we will break that rule, but we'll break it with very particular chemistry. And I would argue you shouldn't need to look at that chemistry, but it's in the competencies, so we'll talk about it briefly. So we've got the alkyne. Cool. What do I want to react the alkyne with? As a hint, It's in the question. Just hydrogen?
Acid?
And let's be a little bit careful. Just a hydrogen? It's H2. Why does that become important? We have H bonded to H. Is that an acidic hydrogen? To be acidic, this one must take the electrons away from the other so that I get H plus, and then I would get H minus. So H plus would be acidic, good, but that only happens if I can get H minus. Is H minus good? No, why not? Being asked to explain why something is good or bad, what should we fall back to? The same thing we fall back to every time. Charge, size, electronegativity, resonance, induction, and oh, I really hope I don't have to answer that. Charge, is it charged? Yes, that's bad, not good.
It's not stable.
Size. Hydrogen is the smallest atom. Is a small atom better able to stabilize a negative charge? No, it's actually significantly worse. So we took the smallest possible atom and we put the most electrons on it we possibly could. Is that a good thing? No. So charge is bad, size is bad. Electronegativity?
None.
It's one of the lowest electronegativities. Does it have resonance? No. Is induction there? No. So nothing about our ranking system says H minus should exist. What does that mean?
It doesn't.
It doesn't. What does that mean?
It's not an acid.
It's not an acid. Good job. The negative of information can help us out. Do you know what Lindlar's catalyst is? No. Okay, so we've just got to work with an alkene and hydrogens. What does the alkene supply? How is an alkene reactive?
Can break bonds, pi bonds.
It is made up of pi bonds, and pi bonds are a source of electrons. So we could take the electrons from this, share with the hydrogen, but if we shared with the hydrogen, we would exceed the octet, so we'd have to kick out H minus. Can we kick out H minus? Is H minus stable? No, so I can't kick out H minus. So that would say nothing that I've done here would work. So I could look for the answer where no reaction happened. Or I happen to remember back to the chemistry that we were talking about, right? And that H minus is a bad leaving group. And if it's a bad leaving group, it can immediately react back, forming effectively a four-membered ring. It was supposed to be in black. We don't care about the ring necessarily. I get a bond to a hydrogen and a bond to a hydrogen. And there's my ring, which we'll call pH. What do I now have? I have an alkene. Cool. What was the point of the word Lindlar's catalyst?
Nothing.
Okay, so we can make the argument nothing. That's fine. You can stick with that for a moment.
Catalyst, like something's gonna happen.
Catalyst would make it happen, yes. So, but the way it makes it happen can have a very important impact on the end result. What is special about the product we've produced? Say it again.
Break the bond.
We did break an initial pi bond. What else is special about it?
It still has a pi bond.
Yeah, so we have an alkene. What's special about alkenes? We could have, we'll come back to that, we could have cis or trans. So remember when we first talked about alkenes, we said, oh, we got to worry about cis-trans isomerism. Yep, cis-trans isomerism is going to come into play here. And it's all going to be dependent on how the hydrogen was added to our structure. Was the hydrogen added where the hydrogens were added on the same side? Or were they added on opposite? Whoops, I can't apparently math. Opposite sides or same side? Will you tell me, based off how we just drew out our mechanism, were they added on the same side or opposite sides? Same side, which means this one's wrong. Okay, and yes, lovely cis. Getting real fancy with it, I like it. Okay, we would get the cis product. Okay, you also brought up an interesting point here. I have a nonpolar pi bond still, which means can I react with hydrogen again?
Yes.
Dang. So I could go through and run this reaction potentially a second time. Fully reduce away the pi bond. Now I'm down to alkanes. Do I have any chiral atoms?
No.
Nope. So do I have to carry about cis or trans or R and S? No, I don't have to worry about any of that. Okay? So this, I would argue, is a ballpark following the chemistry approach. I'm going to delete everything. Can I delete everything? Yep. We can now look at our answer choices. Which of our answer choices align? Structure 2 gives us a cis. Yeah. That was supposed to be colored. Doesn't structure 4 also give us a cis? Why are you saying no, Lindsay? So let's put it this way. Your no is valid, but not to the question I asked. Is it cis? Yes. Absolutely is cis. Why is your no valid?
Because it's
Isn't that an extra?
An extra is a little, sort of, I would argue, still wrong, but your wording and phrasing kinda could work.
Where'd that carbon come from?
How many carbons are in 4? So we're eliminating those two.
5. And the chain?
Does everybody agree with her? 5 carbons in the chain?
Oh,
see
wait.
under the chain.
Okay, good. Everybody's questioning that. That's fantastic. Count the number of carbons total in structure 4, because that'll fix this issue with the chain counting.
Is that full?
Total carbons in structure 4.
Just
And then,
There's 10. Total carbons in structure 2. 10. So your statement of where did the extra carbon come from? There is no extra carbon. That's not the problem.
It's not in the pi bond.
Yes.
It's in the wrong
The
spot.
double bond has changed locations.
Jeez.
So it's not just, oh, well, look at the carbon chain, but you also have to recognize where the chemistry is within the broader sense. Was that really hard to see? Yeah. That's why we did Unit 1, was to give you some practice to help build that so you don't make those mistakes. You're changing the location of where the double bond was, and that's why 4 is wrong. That's why, Lindsay, your no was fantastic, just for the wrong reason. And Sydney, your comment of, well, it has an extra carbon, is wrong, but it's the right idea. right? So finding that difference is really, really critical to being able to approach and answer these questions. Does that make sense? Okay. I think I can actually split screen, so we'll try this. What's the other approach? Split screen, this isn't going to work out well because it's going to make it too small. Do you see the chemical reaction within that table? Yeah? Where is it?
It's like 1 o'clock.
Awesome. Okay. Okay. So that's using H2. You said yes, you see the reaction. Yes, there's H2. But what does it say under the H2? Said another way, does it say Lindlar's catalyst? No. So is that the reaction? No. It's something related to it. But if you had said, yes, that's the reaction, what product would you have ended with? The alkane, not the alkene. And of course, now the other one is slid too far off screen. You would have gained all the way down to an alkane, which we didn't see as an answer choice here. That doesn't mean you wouldn't see that as an answer choice. Do you see the reaction here? Okay. This is why the spider diagrams are insanely challenging. Okay. Even then, you see the triple bond, right? Does that look like the starting material you were given to begin with? And that's up to you to decide. Some people are like, oh yeah, I totally see it. Other people are like, I have no idea. Like, how are you even calling those the same thing? That becomes the problem with trying to memorize all of the reactions. Depending on what thing stuck in your brain, the whole thing can come crashing down. You have to be able to match the pieces to an entirely different, new, exciting structure. Of course, this one's on a split, but it's what's the product? What's the major product of this reaction? So, ta-da, there it is. Everybody think you got it? Mostly figured out? Spider diagram. Is it here? It's a good aggressive shake of your head there, Lindsay. Is it here?
See the picture.
Oh, that is about 7 o'clock. I was like, that's not 7 o'clock. But yeah, I agree with you. Oh, oops. Yeah, there it is. All right, so what does this reaction do? What's the change from reactant to product? Lose pi bond. Say it again.
We have a new OH.
and you add an OH. We good? Based off the information you just gleaned from that slide, yeah, that's exactly what I wanted, Sydney, which sucks. I fully understand it.
Where'd they all come from?
Just get, like, go back to the top.
Oh, I can make it bigger.
Is there 3 oxygens in that? Or no, there's an oxygen in ethanol, too?
Okay. To summarize what you all said, this reagent lost pi bond, gained OH. Admittedly, this structure is obnoxious. I would try to limit your exposure to that. Sort of. This is aromatic, just like the benzene ring, which means it does not react. So, I heard an option that we think the answer is 2. Anybody else want to volunteer a guess?
2 or 3.
Maybe 3. Why did you eliminate 4 and 5?
Changes to the ring.
Changes to that ring structure, which we said should not react. So 4 and 5 should be out. Why did you not say 1?
Because the OH is the it's hard to put into words.
There's a lot going on in 1, but what I would argue that you should look at is where did the OH go? Did it go to a carbon of the pi bond? No. Went somewhere else. So something crazy happened there. Let's not answer crazy. So it's between 2 or 3. You're now down to a 50-50. Are you happy with a 50% on your exam?
Was it 3?
Just gleaning that information, loss of a pi bond, gain of an OH, is not sufficient out of the spider diagram. There was other information embedded in that you did not see. That's why looking at an individual reaction can be very misleading. You would need to understand the explanation behind each of those chemical reactions to explain why a chemical reaction works one way versus another. So just memorizing loss of pi bond, gain of OH doesn't help you out. What else happened? Pull as much information as you can out of that now. Try and remove the other distractions. What changes happened? Try a different way. What kind of chemical reaction happened? Say, yeah.
Addition.
You got an addition. What does it mean to do an addition reaction?
Break a pi bond.
Break a pi bond and
2 sigma bond.
2 sigma bond. Did you say it gained 2 sigma bond? You had to gain something else. What did you gain?
One sigma bond.
You gained 2 sigma bonds. You gained a hydrogen. That hydrogen was also gained to process this chemical reaction, which means you placed 2 things on this, OH and H. Where did you place the OH?
The pi bond that broke.
The pi bond has 2 carbons. Did you place it at either of those 2 carbons? No, it says very specifically only one of those carbons reacted with the OH. The other one reacted with the H. The OH went to the more substituted position. The H went to the less substituted position. If we hold that extra information, what's our answer? The OH goes to the more substituted position. Here's our double bond.
Oh,
We have a green carbon and a red carbon. Which one has more carbons connected to it? The green one. The OH goes to the more substituted, which means this is why it is very challenging to look at a single example, because a single example will not cover all of the information hidden within an individual chemical reaction. You have to be thinking about things at a larger scale, or have come up with other memorization devices to try and nail things down. If you know it's an addition reaction, going back to this, if we know it's an addition reaction, what you could also try and go through and do to memorize, particularly if it's an addition with hydrogen, is that where does the hydrogen go? The hydrogen needs to go to the position that has more hydrogens. So if you know this reaction adds H and OH, the hydrogen must go to the position that already has a hydrogen. The OH must go to the other spot. So there's layers of information that you have to go through and memorize. It's not just the raw, simple fact at the beginning. You also have to process a bit deeper. With that, we have to be done for today, but that's the end of our alkenes. Next week, we unfortunately will get to level it up, and what we'll level it up to, where did they go? Will be our polar pi bonds with nucleophile additions. Just to give you kind of a taste, you get a whole nother system of spider diagrams with more pieces that go along with it. There's a lot of words associated with it. You don't need words, you need structures. Okay, in my opinion. But if you feel you don't understand structures, then you need to learn the words. There's really only those 2 options. Okay, and even more diagrams that come off of it. And at this point, all we're doing is scratching the surface of the chemical reactions that are possible when we're looking at organic chemistry. What I'm trying to set you up with are the tools that you can look at that functional group and predict what chemistries it could react with. So that when you see an alkane or an alkene, you know it's a nonpolar pi bond, it can share electrons. Find something positive. We don't care what it is specifically because you aren't responsible for that in the grand scheme.